Hi, this is a short Q&A video response to our study titled Enological and Species Diversity of Yeasts Isolated from Greenhouse and Vineyard Grapes. And we are 1 Epsilon, the wine research team. Okay, so let's start off with a brief overview of our study by asking the question, what is enology? So enology is the science of winemaking. It's where yeast are used to convert sugars to alcohol and carbon dioxide, and our study focuses on the yeasts that are part of this fermentative process. So different yeasts or different yeast species, they ferment or tolerate different levels of glucose, they produce different amounts of ethanol, and they also produce different flavors and aromas that are found in wine. There are generally 2 populations of yeast species present on the skins of grapes. There's Saccharomyces, and then there's non-Saccharomyces. So Saccharomyces are natural inhabitants of wine cellars. They can convert all glucose to ethanol, and they can tolerate both high levels of glucose and ethanol. Non-Saccharomyces, on the other hand, they are natural inhabitants of grapes. They cannot convert all glucose to ethanol and cannot tolerate high levels of either. But what's interesting is that these indigenous non-Saccharomyces, they provide terroir because they impart unique ester profiles. So terroir is a unique flavor profile that's imparted to a wine by the environment it's produced in. So whether that environment is a vineyard in Italy, or maybe it's a vineyard in the Okanagan, every region basically has unique indigenous yeasts. And these unique yeasts produce wines with unique flavors, fingerprinting the region that they're made in. So it's for this reason that isolation of indigenous non-Saccharomyces is of benefit, because they provide this terroir quality to wine. But isolating a true indigenous non-Saccharomyces is actually difficult, because after the initial harvest and crush that's involved in the winemaking process, sediments or lees that contain commercial yeast strains are often sprayed back into the fields to grow and colonize. So these commercial yeast strains actually decrease the diversity of indigenous species. So the solution then would be to screen an isolated environment for species of yeast and assess the level of diversity. An example would be a greenhouse. Question, which is, what diversity at the enological and species level is present amongst yeast isolates and vineyard grapes? So our hypothesis is that yeast isolates or yeast strains found on the greenhouse grapes will display a higher level of diversity amongst the non-Saccharomyces, whereas the yeast species found on the vineyard grapes will display greater diversity within the Saccharomyces genus. And our approach here was to first isolate yeast from both environments, then do spot assays that would assess the phenotypic diversity, and last we were going to PCR amplify the D1, D2 domain that's present in all yeasts. Okay, so here's the first question we've chosen to answer from our audience. What phenotypic yeast characteristics did you use to differentiate your strains of yeast at the isolation stage? So to answer your question, vineyard and grape berry surfaces harbor complex microbial communities composed of yeast, bacteria, and filamentous fungi. But, as you know, we targeted isolation of yeasts only. So, to accomplish this, we added chloramphenicol to our growth media to inhibit bacterial growth, because chloramphenicol is bacteriostatic. And And at the same time, we also used YPD media, which is yeast peptone dextrose, and that selects for yeast only. A limitation of this method was that we could not specifically select against filamentous fungi, so there was some mold contamination during isolation, but then we were able to eliminate this by increasing the concentration of glucose. So phenotypically, we were also able to visually differentiate yeast from fungi because yeast are generally small, they're creamy textured, form round colonies, and they appear smooth and silky. Fungi, on the other hand, are fuzzy, they're hairy, and they generally have very distinguishable growth characteristics. Okay, so the second question was, what kind of controls will you use for your D1D2 domain gel? Are there D1D2 proteins that have specifically defined molecular weight? So this is a picture of one of our gels showing the results of a PCR we performed on DNA isolated from greenhouse strains. So we used two controls for our D1D2 domain gel. There's the negative control, which is just a PCR tube containing DNA but no primers. We expected no bands, and you can see that in lane 2, we didn't get any. And our positive control is a commercial yeast strain known as Saccharomyces biennis. It's well known to contain the D1D2 domain, and you can also see that in the gel. And the branching question was regarding the proteins. So D1D2 domain, it's a region of the 26S ribosomal DNA in yeast. So there's actually no D1D2 protein per se, but the 26S rDNA, we all know it's the coding sequence for producing 26S rRNA. So the D1D2 domain, it's part of the coding sequence that expresses 26S rRNA. It's variable between different species of yeast, and it's flanked by highly conserved sequences. And so you can exploit the sequences as primers to amplify the region, which is what we've done in our study. So in general, the D1D2 sequence, it's about 560 base pairs in length, and other strains vary in this domain probably by about 10 to 100 bases. And the limitation, though, is that we can't verify that this is truly amplified simply from the gel, which is why we've sequenced our amplified DNA. Okay, so the third question. What other methods are used in assessing phenotypic diversity in yeast, and why did you choose this method? So different yeast strains can tolerate different temperatures, which can affect their fermentative abilities. Measuring temperature tolerance is another method that could be used. It's generally good for yeast to handle a high fermentative temperature. But this kind of assay could have inhibited other strains during our screening, which is why we didn't use it. A hydrogen sulfide test would have been another method because one of the characteristics of good wine yeast is little contribution to wine faults during production, which often lead to wine spoilage. And one of the most commonly encountered wine faults is production of hydrogen sulfide, which causes a rotten egg smell found in some spoiled wines. So for a method like this, you would make plates containing sulfite, spot serial dilutions of your culture, then if they produce hydrogen sulfide, the colonies change color from white to brown. And that would be a hydrogen sulfide test. And last, another common method used is fermentation assay that's quantified via HPLC, which stands for high performance liquid chromatography. So in our study, we showed phenotypic diversity by fermentation of glucose, production of ethanol by spot assays, but HPLC is much more quantitative, and it provides the same key information in assessing the phenotypic diversity, but with respect to applications in wine production in a more quantitative way. So it's definitely more useful. And definitely because of time constraints and unavailability of specific reagents, we were unable to pursue these routes. But there are definitely even more methods out there for assessing phenotypic diversity. For example, you can do an amylase test to identify yeast strains capable of producing amylolytic enzymes, and that's also of benefit to winemakers. And I believe that answers that question, and that's a wrap. So we sincerely hope you enjoyed watching this video and that this has answered your questions. If you have any further inquiries, feel free to reach us via email. We are Brittany Goldhawk, Jeremy Lotto, and Manjot Kilon from the Department of Microbiology and Immunology at UBC. Thank you.